Hi class, we're going to continue with the chapter on blood, and this lecture is going to go over blood types and disorders that are related to blood types, so issues that occur with blood typing. We're going to start with talking about the 4 types of blood, and I'm actually going to try to draw and diagram this out for you, and then we'll take a look at the PowerPoint lecture. I'm going to attempt this. It's always kind of awkward and weird because the way my camera is and everything, but we might be able to make this work. If not, I'll just get a good arm workout holding this whiteboard up. So there you go, put my leg up there. There are 4 types of blood. So you have blood type A, you have blood type A, B, you have blood type B. Actually, let me do this. You have blood type B, that might make a little more sense. You have blood type A, B, and you have blood type O. Now, when I look at this, I see a reflection. So I don't know if you're going to see this backwards or not. Hopefully you don't. All right. So in blood type A, well, let's first talk about why these blood types exist. So these blood types exist basically because there is a specific marker or antigen on your blood cell. So A blood has A antigens on it. B blood has B antigens. An antigen is a marker or a special extracellular protein. Okay. B blood, guess what that has on it? A's and B's. And guess what O blood has? Doesn't have O antigens. It has nothing. It's like the naked cell right here. Okay, so those are the antigens that are on these types of bloods. Now, the reason these antigens, well, not really the reason these antigens exist, but what happens with these antigens is they can produce an immune response, right? So say, for instance, you're invaded by a bacterial cell. Well, bacteria have antigens on their cell membranes, plasma membranes. And so what happens is our immune system recognizes those antigens as foreign and says, oh, you don't belong here. I'm going to destroy you and get rid of you and eat you into a million pieces or whatever. So antigens can stimulate an immune response. The immune response starts with antibodies, right? So antibodies are in response to antigens. People that have blood type A will produce B antibodies, right? They're anti-B, they're A, right? People who are blood type B produce A antibodies. People that are blood type AB don't produce any. People that are blood type O, guess what? They produce A and B antibodies. All right, so now it gets tricky when we talk about who you can give blood to and who you can get blood from. Give to. So people who are blood type A and produce B antibodies can only give to other people that have the A antigen. So they can give to people who have A blood or A B blood. They're not giving anything to this person that they don't already have. Right? People who have B blood are anti-A, so they can give blood to only other B people or to AB people. People who are blood type AB can give to other AB people. Can they give to A? No, because A produces anti-B and AB has B, right? So AB people can only give to other A people. How about blood type O? Blood type O has A and B antibodies. So guess what? Blood type O, which doesn't have any antigens, can actually give to everybody. Blood type O, you're not giving anything extra to A. There's no O antibodies here. It's the naked cell, remember? So people who are blood type O can give to A, can give to B, and can give to AB. How about we'll say can receive from. So who can you receive blood from? If you are blood type A, you can receive blood from other A blood. Can you receive blood from AB people? No, because you're getting a B, right? If you're blood type A, you can receive blood from other A or other O people. If you are blood type B, you can receive blood from other B people or other O people. If you're blood type AB, you can receive blood from everybody. You can get it from A, you can get it from B, you can get it from AB, and you can get it from O. It's called the universal recipient. Universal recipient. And then blood type O people, oops, we left off here. Blood type O people can give to other O people. I should have said that before. They're called the universal donor. So other O people can receive blood from only O people. So O people can give to anybody that can only receive from other O people. AB people can only give to AB people, but can receive blood from anybody. Blood type A people can only receive blood from A people or O. Blood type B people can only receive from either B people or O people. So there's your visual. Again, hopefully it's not backwards for you. If it is, use a mirror. I don't know, to figure it out. Sorry about that. So let's chat about how all this works together. And so we're going to switch to the PowerPoint and kind of go from there. My arm hurts. It's a little bit of an arm workout. So I reviewed this already for you, but this is it in words if you prefer words as opposed to my babbling over a whiteboard. Blood types are determined by antigens. As I said, they're those little cellular markers. They're genetically determined, so they are inherited. And these are going to trigger an immune response. So they're basically identifying the blood cell as, yes, I belong here, or no, I don't belong here. So there's actually lots of different surface antigens on our blood cells, but the ones that we are concerned with are the A and the B, and I didn't mention this, but there's also a D antigen. The D antigen is that positive or negative. So if you know your blood type, me, for example, I'm B positive. That means I have the B surface antigen and I have the D surface antigen. So that just makes my chart not look so pretty when I add the D antigen in it. So I don't put that in there. But the D antigen is very easy to understand, and we'll talk about it as we go through. So it's an extra something. So these blood types are based on those surface antigens. The surface antigens are also sometimes called agglutinogens because they cause agglutination. Agglutination is the clumping that occurs when there's an immune reaction. So blood type A only has A antigens, B only has B antigens, so on and so forth. I already went through this in my little chart. There they are, a much better picture than what I drew on my wimpy chart. The antigens and the antibodies that are created. Remember, AB blood does not have any antibodies, and O blood has all of the antibodies. So let's talk about this process of agglutination that occurs. Agglutination is a process of clumping. In blood, it's going to occur when the surface antigens, or agglutinogens, are exposed to the antibodies, or the agglutinins, right? So when the immune system matches up with those foreign antigens, they're going to clump around it and cause this immune reaction. So an example of when this would happen would be like if you give type A blood to someone who has type B blood, right? Mixing those blood types. It's called a cross reaction. Those cells are going to undergo hemolysis, right? Which is the rupturing of the red blood cells. So the red blood cells are going to rupture, burst, can cause dangerous clumps and fragments within the blood. It could damage the blood vessels and destroy tissues because if the blood vessels are blocked, that means blood can't get through. So there's the process of agglutination that's occurring and then hemolysis that occurs afterwards. So that Rh antigen or D antigen is named after the rhesus monkey, which is where these were determined. And this is going to either exist or not exist on your blood cell. So you're going to be positive for it or negative for it. So if you have O negative blood, that means you have a completely naked red blood cell, does not have any antigens on it. If you have an O positive blood, that means you don't have A or B antigens, but you do have this D antigen. And the same rules apply. So you can't give your blood to somebody that doesn't already have those antigens. So an O positive person, even though O is the universal donor, an O positive person cannot give to someone who's AB negative because someone who's AB negative does not have that Rh factor or that D antigen. Rh factor, D antigen, same thing. So O positive people can only give to other positive people. An O negative person can give to anybody because they don't have any antigens. So it's interesting to actually see and observe this, but there is actually a correlation between where your family history is, where your family is originally from, and your blood type. So the majority of people on the planet are type O blood. It's the most common blood type. But people who have a Asian heritage, either Chinese or Filipino, Korean, Japanese, tend to have a higher occurrence of AB blood. AB blood is the rarest in the world, but people of Asian descent are more likely to have AB blood. People, and likewise, same thing with type A blood and type B blood. It's more common in different lineages. People of Native North Americans tend to have A antigen. People of South American heritage do not. Almost all it says here in the chart, 100% of South American natives have O blood. So, and same thing with RH. RH is much, much more common than to be Rh negative or to have a negative blood type. So let's talk about how we know, and we're going to do one of these in lab if we haven't done it already. So the blood typing test, you're basically mixing the blood with different antibodies and observing for clumping, or agglutination. So agglutination is going to occur where there's a corresponding antigen. So type A blood is going to clump with the anti-A antibodies. So this is necessary so we make sure that we avoid transfusion reactions. You have to have a donor and recipient compatibility so there's no cross-reaction. And so this is something that you will see in a lab setting. Our test is similar to this that we'll do in lab. So if you look here, there was clumping. See how it's clumping? It's not like a nice smooth blue. There was clumping here in the anti-A category. That means that this person has the A antigen. There's no clumping here in the anti-B, and there's clumping here in the anti-D. So that tells us that this person has A positive blood. Let's look here. There's no clumping here. It's a nice smooth kind of bluish green color. There's clumping here that occurs, and there's clumping here that occurs. This person is B positive. Here there's clumping in all three. So this person is AB positive. Here there's no clumping at all, so we have O negative. So that's what you're looking for when we do these blood typing tests in lab. You're looking for clumping. Clumping means a positive test. So let's talk about the genetics of blood type. The presence of anti-A or anti-B antibodies is actually genetically determined. It remains constant throughout your life, so you'll never change blood types. There's no need to be exposed to foreign red blood cells to develop those antibodies. They're there already. Anti-Rh antibodies are different, though. They're not automatically present. So an Rh negative person does not automatically have anti-Rh antibodies in them. If you have so let me explain again. If you have blood type B, you naturally already have anti-A antibodies in your body, right? But if you're Rh negative, you don't already have anti-Rh antibodies. You're not born with those. You have to have what's called an exposure. So once you've had an exposure to Rh positive blood, you will then develop those antibodies for it if you're Rh negative. So there's only a few examples where this is important, and that's what we're going to talk about. After you answer these three questions, pause the video here, answer these three questions, make sure they're all sunken in. I'm going to keep going. So let's talk about this clinical case called hemolytic disease of the newborn. So as I said before, blood type's determined by genes, and if you are a type A blood, you're naturally born with anti-B antibodies. But if you're Rh negative, you do not have anti-Rh antibodies. You have to have an exposure incident. So anti-Rh antibodies can actually cross and attack fetal red blood cells. So if the blood type of the fetus is different than the blood type of the mother, there could be some issues. And hemolytic disease of the newborn, this is a condition where the maternal antibodies are crossing the placenta and attacking fetal red blood cells. Has many forms, a variety of severity. It's most common, though, in mothers who are Rh negative and a fetus who is Rh positive. So if you're female and you're Rh negative, or you don't know your blood type, get tested to find out if you're Rh negative, because this could help explain infertility problems or problems with miscarrying. So in a mother who has, this is their first pregnancy and you're Rh negative, there's very few things to worry about. So during pregnancy, very few issues will occur if you're Rh negative, because, as we said, you're not born with those anti-Rh antibodies. Unless you received a transfusion that was wrong at first or whatever, you would probably know about that. You would never have been exposed to Rh positive blood. So very few cells enter or are exchanged during the actual pregnancy. So the immune system actually isn't triggered to build those anti-Rh antibodies yet. You have to be exposed to the Rh antigens in order to develop the antibodies. So an Rh negative mother doesn't have those natural antibodies. As we said, you're not born with them. But at birth, there is some mixing of the placenta and the uterine blood. It's just from natural detachment of the placenta, etc. So there's some exchange. That's going to expose the mother to the Rh antigens. So the mother now has Rh antigens in her body. She can produce anti-Rh antibodies. It's a process called sensitization. So about 20% of Rh negative mothers who carried an Rh positive child sensitized within 6 months. So in other words, a decent percentage. The first infant's not affected because there's no antibodies, right? You have to be sensitized through an exposure incident to create those anti-Rh antibodies. In a subsequent pregnancy of an Rh mother with an Rh positive infant, those anti-Rh antibodies can cross the placenta, go into the fetal bloodstream, destroy fetal red blood cells, cause anemia, dropping fetal red blood cell count, dangerous levels. It's called erythroblastosis fetalis. So in other words, there's very high fatality rate if there's no treatment. Newborn can be born anemic or jaundiced. And the maternal antibodies remain active for several months. You could end up with a newborn that requires an entire blood transfusion. Hemolytic disease of the newborn can be prevented by RhoGAM. It's just a medication that decreases sensitization of the mother's antibodies. So with that mixing, it's not going to attack. So here's the whole process. With the first pregnancy, again, there's no real concern with the first pregnancy unless she's had an exposure incident already. An Rh negative mother with an Rh positive fetus. There's no mixing yet until delivery, when there's some mixing, and then the maternal sensitization process happens, and the mother, Rh negative, can now produce anti-Rh antibodies. So with the second baby, she's now sensitized to the Rh antigen and can attack, her blood can attack the baby's blood. Her antibodies, I should say, can attack. It's the whole process in sequence. So that's hemolytic disease of the newborn. You can pause the video here, go back, answer these 3 questions. Actually, you don't even have to pause it. You know what? I think I'm going to stop here. And we are going to have another video which will cover our white blood cells.